On behalf of the Biochemical Society and Portland Press, I'm pleased to welcome you to today's webinar, which is part of our Biochemistry Focus webinar series. Topics in the series include different research areas in the molecular biosciences, as well as practical sessions to support career development. Each webinar will give you the opportunity to ask questions via text, and we welcome suggestions for future topics and speakers to feature in our webinar series. Please see the website for more details. So I am Femi Olaroniji, reader in synthetic biology at Liverpool John Moores University. My research interest is in the structure, mechanism, and application of large serine recombinases, and I am a member of the Biochemical Society Awards Committee. In this webinar, we will hear from the Biochemical Society's 2025 AstraZeneca Award winner, Professor Róisín Owens from the University of Cambridge. Before I hand over to Róisín, I should mention that questions will be asked after Róisín's presentation, but you can send in your questions during the talk. If you have a question, please type it in the question box as shown in the image on the screen, and we will try to answer as many as time allows. So Róisín is Professor of Bioelectronics at the Department of Chemical Engineering and Biotechnology in the University of Cambridge, and is Deputy Head of the School of Technology, in charge of research and strategy. She received her BA in Natural Sciences at Trinity College Dublin and her PhD in Biochemistry and Molecular Biology at University of Southampton. After this, she carried out 2 postdocs at Cornell University on host pathogen interactions before becoming a group leader in the Department of Bioelectronics at École des Mines de Saint-Étienne. Róisín has won several awards for her work, and currently her lab focuses on the integration of electronic devices with biological systems for continuous monitoring, with a particular interest in studying the microbiome gut-brain axis. So Róisín, you have our full attention.
Thank you very much, Femi, and thank you to the Biochemical Society. I'm very pleased to be giving this webinar today. Just to confirm, you can see my full screen, okay?
Yes, I can see your screen.
Wonderful. So actually what I put on the first screen is just a snapshot of the recent article that I wrote, which has been published by Portland Press. And it was a really nice opportunity to reflect on my career path. I started as a biochemist, and I then migrated into biomedical engineering and then electrical engineering. And I feel I've come full circle now, because I hope that the technologies that we've developed have become useful to biochemists and other biological sciences. So I'm going to tell you about our work on the gut-brain axis, where we're using electronic devices to do what I hope are interesting things. But don't worry, I'll give quite a lot of introduction into the various electronic methods as I go along, and also the types of biology that we're accessing. Okay, so I'm fascinated by gut health and the gut-brain axis. And this dates back to my PhD when I was working on enteropathogenic E. coli. I was working on protein structure and function. Back then, enteropathogenic E. coli, as the name will give you the clue to, they're pathogens. But even, we thought of bacteria as being maybe not so good. And we didn't really know so much about the microbiota and the positive effect that they could be having, and the fact that our sedentary lifestyles and bad diet might be leading to us wiping out some very important parts of our ecosystem. So what surprises a lot of people is the fact that in various neuropathologies, some of the first symptoms are perceived in the gut. So for example, people with Parkinson's disease, one of the first symptoms they may experience is difficulty in going to the toilet. This is an altered motility. We know that people with anxiety may have gut dysfunction. People with post-traumatic stress disorder, around 30% of those people may have some kind of disturbance in their bowel habits. So there are many gastro and neurodegenerative disorders that are linked to poor gut function, but these disorders aren't really understood. And diagnosis is hard, but also treatment is hard. And we actually really need just a lot more fundamental understanding. And for me, a functional measurement measures the function of the gut, measures how it works in the normal circumstance, and then you can compare that with a disease circumstance and be able to tell what it is that's working differently. We're particularly interested in 3 functional measurements. So we have spent a lot of time looking at the permeability of the gut, and I'll explain that. We've more recently looked at gut motility, and probably the most difficult, but maybe the most interesting, is the enteric nervous system and how different components of our immune system and our enteroendocrine system may be affecting our brain health. So it's complicated. The environment in the gut is consisting of many, many different moving parts, and the connection between gut and brain is not that well understood. But what we know is that we have this very complex interface. We have a mucosal lining. These are typically mostly epithelial cells, although of course you've got goblet cells and these enteroendocrine cells facing into the lumen of the gut, where you've got trillions of microorganisms. Then you've got a connection that could be going, for example, you've got soluble metabolites that could be produced or co-produced by microbiota that are crossing that mucosal layer and somehow getting into the bloodstream. You could have the enteroendocrine cells or other cells producing hormones, neurotransmitters, which may be affecting local nerves or going maybe via the vagus nerve. We know that there can be an immune cell component where immune cells may be picking up signals locally and then potentially transferring or traveling to the brain. There was a recent publication in Cell saying that local macrophages in the gut may be responsible for accumulating some of these early types of plaques that are found in Parkinson's. So very complicated and very difficult to understand without a huge amount of additional study. There has been limited, I would say, electrophysiological intervention in terms of the gut. If you look at most other parts of the body, there are electrical devices used in clinics like your EKG or electrocardiograms or electroencephalograms, all kinds of grams used to measure different things, but somehow in the gut these are not used in the same way, even though you have lots of electrical signals that you can measure. And so this particular paper was highlighting that technology for diagnosis of GI dysfunction is underdeveloped and that EFIS methods were not finding widespread clinical utility. So that was sort of the call to action. Now, if I explain a little bit about what I understand by bioelectronics, so most people think of bioelectronics, if they have any conception, as the interfacing of a cell like a neuron with an electronic chip. And there they think about the chip as being, you know, a typical silicon chip that you might find in your smartphone. And typically the cell is thought of as a neuron because these are known to have action potentials which have been recorded for centuries. So those two things combined gives us bioelectronics. And in fact, there are examples of bioelectronic medicine, as I alluded to just a while ago. I said that there are all kinds of things that are happening where mostly stimulation is done, where you can stimulate a lost function, for example, sacral nerve implant to help with incontinence. And then, of course, you can do the recording that I mentioned too. So that's typical bioelectronics, but actually bioelectronics can be a lot more. And if you remember back to your textbook, cell biology, you'll remember that the cell membrane, so surrounding mammalian cells, you have this highly insulating lipid bilayer, so it doesn't even allow charged ions to cross through, except that you've got these transmembrane proteins inserted, so you can get modulation of ion flux. And the cells work very, very hard to maintain differential concentrations of ions inside and outside the cell. Anytime you've got that kind of a barrier with differences in ion concentrations, you generate membrane potential. And in fact, measurement of membrane potential is a very good sign of, is a cell alive? Because if you have no membrane potential, typically the cell is dead. Now that's true at the level of the cell, but it's also true at the level of tissue. So you can see an example here of cells that form tight junctions. It's a typical barrier tissue, also found in the gut. And so being able to measure the restriction caused by those cells in terms of ion flux is also very interesting. So you can measure membrane potential, you can measure ion flux, and that means that all cells have the ability to be accessed using electronic devices, because electronic devices will change based on the environment created by ions. And I'll explain to you how electronic devices can be used to very sensitively measure things like membrane potential and things like barrier function. Now, in terms of what's happening now in the literature, we're seeing a real uptick in people developing electronic devices for gut applications. So the promise that was mentioned in the article I alluded to has started being seen, but it's not in the clinic yet. It's being seen in research labs. And so you've got some examples where people are developing mostly intraluminal devices, so either an ingestible pill, which could maybe do some sensing, so that's shown on the right, or you got these examples of fiber photometry. I won't go into the details, but you could do optogenetics, for example, if you have a fiber that can go down into the gut. There are other people who've developed rolled up multi-electrode arrays to measure those typical electrical responses, say from neurons or muscles in the gut. But it's complicated, and the technologies developed for CNS may not work very well, so for the central nervous system, for the enteric nervous system, because the structure is so different. So there's still a lot to do, and although it's great that there are people in this area, it just needs a lot more, so definitely more to be done. This was an interesting paper I saw recently, a mini endoscope that could be used in lab animals to try and hook sensors onto an endoscope. So you could imagine that then could be worked into a clinical protocol where a gastroscope could be used to get more information. So rather than just imaging tissue, you could maybe record some electrical signals. Okay, so I'm not getting into the detail in any of those things, but it's just the bottom line is that we're seeing much more engagement with the idea of functional measurement in the gut. So when I started thinking about the gut-brain axis, I was really thinking about what sorts of samples we'd be able to get and how we would do functional measurements. And really up until fairly recently in my group, we'd been developing in vitro models. So looking at development of these new microphysiological models where we try and have many cell types, we try and think of the environment in terms of gases, 3D nature, gradients, fluidics. How do we recreate things so that our in vitro model is as physiologically relevant as possible? Now, the nice thing about in vitro is the models are human and they're high throughput. If you go through the line of, say, drug discovery, then you get to, well, you could get ex vivo samples, you can still get human samples, although they'd be rare, they'd be biopsies, but then you can start entering into the world of doing animal experimentation, either using ex vivo samples or in vivo. The relevance may start to increase systemically, although the animal experimentation is a problem. But there's a real limit to what you can do on humans. You can get blood samples, you might be able to do behavioral studies, but then of course the throughput goes down. So what you'll see in today's talk is that I'm going to focus, our newer work is more on the in vitro side, because having shown proof of principle for a couple of other devices in animals or using ex vivo tissues, we think we still have a lot to learn in the human in vitro model, but we have to make those more representative of that very complicated environment I talked about in the gut. So what I'm going to do is tell you about 3 functional measurements using 3 device types, all based on a similar technology, but measuring 3 different things. So we want to measure gut permeability of the mucosal layer in the gastrointestinal tract. We want to measure gut motility, where this is that contractile ability of the gut, which is part of the autonomous nervous system. You don't have to think about it. The gut contracts and moves and mixes food as it goes down. And then we've also got the enteric nervous system, So the motility is part of the enteric nervous system, but there's also the function of the gut as a sensory organ where it reacts to and senses food antigens, but also signals from hormones and neurotransmitters, and then transmits that to the body, for example, via vagus nerve or via the circulation. So being able to access 3 functional parameters using 3 device types. So we're going to start with the in vitro part. So there we're trying to build up this complicated model with some components, and of course we can't do everything. And so some of the more complicated layers, like the muscular layers, very difficult. We can have representative cell types. And of course, if we use things like organoids, we can get more cell types. But you'll see that we have the ability to ask specific questions, which allows us to have simpler, more robust models. But for some questions, we have to have more complex models. And then sometimes we need to move to ex vivo or in vivo for questions that we simply cannot answer in vitro. Okay, so the first thing I want to tell you about is it's a model of the mucosal layer in the gastrointestinal tract. Typically, we're making either single layer or it could be multi-layer cellular structures, so they're more than one cell type. We're typically trying to integrate things like hydrogels, so we have a more 3D structure. And then just a note to say, we mostly grow our mucosal cultures in something that looks like a transwell, where you have a suspended insert, so hopefully you can see my pointer, so that the cells perceive cues from above and below, because very well known to be important for their differentiation. So I'm going to work through a couple of examples, but I'm going to introduce you to the technology before I go any further on the cell biology. Okay, so my first foray into electronics, so I showed you a picture of a chip, and it was a cartoon of something that looks like a silicon chip that's hard and rigid, and it's very 2D. And the reason I got interested in electronics back when I was a postdoc was because I heard about a new type of electronics using polymers. Now these polymers, we use one, it's a mixture of 2 polymers actually, P dot, it's a type of thiophene, and PSS, which is a polyelectrolyte. And these come as a liquid formulation. So what that means is you can make them into all kinds of shapes and sizes. If you look at the chemical structure, it's not that dissimilar from some of the types of molecules you might see in the extracellular matrix. It is a mixture that becomes extremely hydrated because of the high amounts of charge, so like glycosaminoglycans. We get much, much better interfaces with the biology because we don't have so much of a mechanical mismatch. So we know if you were to implant a device, not what we're doing here, but same principle. If you implant a device that's very rigid in tissues, the tissues can reject it and wall it off in a way. If you get closer to the tissue, that results in higher fidelity recordings. And there's a property very important in electrical recordings, which is impedance. If you have what are characterized as low impedance materials, then you get a better signal to noise ratio. So this is very important. A final thing, I mean, there are many reasons I like these materials. They're also optically transparent, by the way. You can mix them because you start with a liquid formulation. You could start doping in materials like collagen or hyaluronic acid, which we've done in the past, to make them a bit more biomimetic or maybe a bit less stiff. So we've made lots of different device types. We could make the traditional 2D arrays, but I've been focusing more recently on, so the first example I'll show you is using flexible conformal devices that are super lightweight, so in the order of 1 to 2 microns. So they sit on top of tissues and don't damage the tissues. The tissues are fine and healthy, and so you can do chronic experiments without issue. So the type of measurement, if any of you have ever looked at measuring resistance of barrier tissue, so that's that resistance that you measure when the tissues are blocking ion flow from traversing through. It's a well-known characteristic of barrier tissues. In many labs, you'd see what are known as chopstick electrodes. So those are stainless steel electrodes, and one goes on either side of that transwell insert, and you measure the resistance across the tissue. If it's a high resistance tissue, blocking ion flow and blocking lots of other things from going through, then you measure high resistance. If, for example, you had a pathogen or something, a detergent that opens up those cells, then you measure a low resistance, and we would see that easily with the chopstick electrodes. There are many problems with the chopstick electrodes, and so we like a device type that can conform better to the tissues. Chopstick electrodes are scenario A, where you have an electrode on either side. Our electrodes typically sit below the tissues or above the tissues, and so we get a very similar measurement. We don't get a single frequency measurement. So if you're doing chopstick electrodes, you measure at a single frequency and you get a single value. We get what's called electrical impedance spectroscopy, broad spectrum of frequencies, and we can actually tell different things from the different frequencies. I won't go into that more today, but suffice to say that to measure the transepithelial resistance, we want to be in this mid-range of frequencies, and that's very well known. We're not the only ones doing this. There's a whole field of electrical impedance sensing, and we get this very characteristic shift in the mid-frequency range when we have barrier tissue in contact with our electrodes. And you can, if you know a bit about electrical engineering, you know you can apply circuits and you can extract different parameters. But essentially, mostly what we're looking at is this resistance of the paracellular junctions between cells. Okay, so we've got an in vitro model of the gastrointestinal tract, which is in one of these transwells. And I'll work through how we make that model. And then this is our conformal device. So each of those round spots here is a different electrode. And then this is a flex cable, which we then connect to our box, which captures the signal. And so if I show you the data here on the right, what we've got is the baseline, which is the bare electrode. When we take our electrode and we place it on top of tissue, we expect to see a shift upwards in this mid-frequency range. And I can go through the data in a second. We extract the resistance, which is this resistance here. We can do that in vitro on a transwell, and we've actually shown we can do that on pieces of tissue ex vivo in an organ bath. Okay, so the in vitro model of the gut, it's using cell lines, just to be clear about that. And because we want it to be a bit more representative, what we've got here is a hydrogel which we made out of rat tail collagen. We embed macrophages in there, so monocytes that we've differentiated. And then on top, we place two types of cell, KCO2s, which are colonic enterocytes, and then goblet cells, which produce mucus. So that protocol develops after about 3 weeks. We have differentiated tissues, and we've checked that using standard electrodes. One thing to point out, though, is that these gels sometimes contract, which makes a standard electrical resistance measurement with an electrode above and below very difficult, because all of the ion flow goes on the sides where the tissue is pulled away. That's not a problem with our device because it sits on top of the tissue. So we had previously shown we could do this with monolayers. We place the device on top. You see the baseline, you add the cells, and you see the shift, and then you can extract the resistance. And then you see the same type of signal if we have our more complex model baseline compared to the cell culture. This is a triculture barrier. Now we can use a functional assay, which is called a calcium switch assay. So you need calcium for those paracellular junctions. And so if you add a chelator of calcium, EGTA, you remove the calcium and you lose that resistance. Now this is a dynamic property, so if you replace the calcium, you gain back that. And that's what we're showing here. After we've added EGTA, we drop our signal and then it recovers if we add back the calcium. And we see the same with the triculture model. So that's proof of principle that it works. And this is the comparison where we've done a similar measurement using the same device on a piece of tissue from mouse small intestine. Mouse small intestine we would expect to be less resistant than our cell lines because they're representing colon, but we still see a signal, and we do our EGTA assay and we see disruption and recovery. Okay, so we have been working with some gastroenterologists at Addenbrooke's, the hospital in Cambridge, and they had previously studied IBS patients, so irritable bowel syndrome, and they had been able to categorize the patients into two groups based on sequencing of their microbiota, where they discovered they could characterize a pathogenic microbiome versus a non-pathogenic microbiome. Now, both groups of patients have IBS. It's just that one is associated with, the pathogenic one was more associated with diarrheal disease. They could then use this FODMAP diet. You can see the acronym explained there. It's a very bland diet where if you went on this diet, it could transform your microbiome from the pathogenic-like type to the more health-like type. And so this could be a way of treating some of these patients. So we had access to fecal samples from a subset of these patients, and the idea was to check if we could have a functional measurement of those fecal samples. Now, the fecal samples may contain live cells or cells which can be recovered because they store at minus 80, but we were just interested in the metabolites that might be present. And so we would take the stool samples, resuspend in PBS, filter them, and then add the filtrate on top of our model. Now, our model is a healthy model. We're not looking at the tissue of the patient. So we're separating the, if you like, pathogenic microbiome from the whatever maybe underlying genetic component could be associated with the tissue. So we're just looking at that microbiome and seeing, would it have any effect on a tissue model in terms of increasing or decreasing the resistance. And so we would measure metabolomics, measure cytokines, because don't forget we've got those macrophages in there, and then also measure the resistance using our conformal device. So we had 5 households. The reason household is important because we've got the patient and we've got their household control. So somebody who does not have IBS, but who would have the same diet and possibly a very similar microbiome. So those are the grouped samples. And we had IBS-P, so based on previous sequencing, and IBS-H. And what we see is that in 3 of the households which have this pathogenic microbiome, we see a difference in the resistance between the patient and the household control. Now, it's a difference, all of the resistances go up, right? So this is our healthy tissue model, but don't forget it's lacking a lot of components. All of the resistances go up. They go up a lot more for the household control than they do for the pathogenic patient. And similarly, in those same households, we tended to see an increase in one cytokine, IL-8. We tested others, but IL-8 is known to be the sort of pro-inflammatory cytokine. You can see on the left here the severity score of the patient. So the higher that score, the more severe their disease is. This is based on a questionnaire. Following the FODMAP diet, which not all the households that we had selected did, only 2 of them did, we see that that difference in resistance has gone away. So it's evidence that our resistance is a proxy for the sequencing in terms of being able to tell whether the FODMAP diet might actually work. Okay, we're continuing to work on that. We're identifying some interesting metabolites in those fecal supernatants and doing more samples. We have a new trial that started, and so we're recruiting newer samples from there. Alex Wheeler is the PhD student who was doing this work. She wanted to make her model even more complicated, so she decided to include also endothelial cells. So you can see the experimental scheme here. She's using Huvex, again, you know, nothing very fancy in terms of the cell type. What's good or more complicated here is getting those 4 cell types to grow together and to be able to do the assay. So Alex reasoned that you would be able to measure not just the epithelial barrier, but also be able to measure the endothelial barrier. So this is just showing that she's confirmed the presence of the 3 cell types. So she's imaging on the top, in the center, and on the bottom. If you take the chopstick electrodes, so this is an illustration of those, you measure a bulk response if you measure the quad culture. So you measure a transepithelial resistance. But you don't really measure anything from HUVECs. They're known to be quite leaky, and the signal to noise of this device is just not high enough to be able to measure anything. Whereas if Alex took her device and places it on top of the barrier, then she flips the transwell and measures it on bottom of the barrier, she can actually measure 2 different barriers. Now you see here, this is the quad culture in the red line, and then the HUVEC barrier shown in the blue line. Now those of us who are used to looking at these kinds of plots can see that those lines are quite different. We can characterize differences probably in the capacitance. I won't go into that more now, but just to say there's more data you can extract because we're doing a sweep across all frequencies. And that's really the beauty of electrophysiology, is the richness of the data. The downside is, how do I interpret this? How do I know what it is I'm measuring? But so this is a new type of data that we can get from these models. And the way she tested this was, so now not using fecal samples, but actually using combinations of different bugs which are known to be present in human microbiota. To cut a long story short, the major thing that she noticed was that individual bugs didn't have that much of an effect on the resistances of our tissue model, but a combination of the bugs, so this was a community of actually 20 bacteria, it seemed to have a significant effect on increasing resistance. So, you know, if you were to think, how is this useful? Well, what combination of probiotics might I take that would give me an increase in resistance? And in case I didn't say it already, increased resistance in gut barriers is seen as a good thing because it reduces the number of compounds or of pathogens or other things that might transfer into the bloodstream or elsewhere. So the gut is supposed to be highly resistant, and it's thought that the more resistant it is, the less undesired effects you have from things present in the gut. So that's just a general principle. People with high anxiety who have gut issues may tend to have a high permeability in their gastrointestinal tract. Okay, so we're improving these models because, as I said at the beginning, we need to be able to understand more about the gut-brain connection. So we've been working at this mucosal interface, but really we need to include enteric nervous system cells and other cell types. So that's an ongoing project. We're currently developing a protocol to differentiate enteric nervous system neurons. It's a challenging project, but we're persevering. But the other thing we're doing is using patient-derived organoids. So I'm just going to tell you about a little bit of work we've been doing recently. It's a slightly different device, so I'll explain that. But just to say, so this is the workflow. So we're working with Matthias Silbauer, who's a pediatric gastroenterologist at the Stem Cell Institute here, and Róisín is a PhD student who's been working with us on this project. So Matthias has built up this amazing bank of organoid lines from patients, human patients clearly, where they can do biopsies and then isolate cells. They've done a huge amount of work on the sequencing of these. But then Róisín's project has been trying to culture these in vitro. Now, actually, we don't just have the epithelial organoid, they've got matched mesenchyme samples so that those can be used also in the models. And then the idea is that you could have a way of challenging these models and looking at different factors. So here we have a different project in the lab where we're not using conformal devices, we're actually making three-dimensional scaffold devices. So these are scaffolds like extracellular matrix scaffolds that you might see used for tissue engineering. But instead of an extracellular matrix sort of passive scaffold, we're actually making these out of conducting polymers. And then we are introducing organoids and mesenchyme in there. So again, back to my devices. So this is this kind of device here, a 3D scaffold. And how we make these into cell culture devices. So we freeze dry these and we're left with this porous sponge. And then we slice them, we integrate them into well plates. And so we've replaced essentially the filter in the transwell. And we have a fairly high throughput system for doing electricals. Now we have got the hosting of the cell and the monitoring of the cell culture into the same device. So it's a bit cyborg, but it's highly effective for continuous monitoring, not just the measurement once you've made the model. So this is how it goes. We see devices. We use a type of fibroblasts. These are cell lines, just to show you how the experiment goes. We see the fibroblasts first, then we add the intestinal epithelial cells, and then we mature. You see the type of data that we get is very similar to what I showed you with the conformal device, a shift in the mid-range frequencies that occurs over time as the cells differentiate. So the protocol with Róisín was different. So he was taking biopsies either from the terminal ileum, so small intestine, or the sigmoid colon, large intestine, and then dissociating those after they were grown as organoids. So he also has the matched mesenchymal cells. He seeds those in. So this is in place of the fibroblasts. And then 5 days later, he has the epithelium. These types of cells grow a lot faster than our cell lines. So after about day 7, we see differentiated epithelium. But the difference now is that we have many other cell types that we never would have had in our previous models, importantly, enteroendocrine cells, stem cells, and TA cells. Okay, and then just showing the difference between SC and TI in terms of the type of cell we would expect to get. He's done staining to show the type of markers you'd expect. So mucin produced by the goblet cells, LGR5 is the stem cell marker, occludin for the enterocytes, chromogranin A is the tough cells, no, sorry, the enteroendocrine cells, or I may have got that wrong. And then EpCAM is the general epithelial marker. So can we measure? So here we've got the terminal ileum and the sigmoid colon. We expect the terminal ileum to have a lower resistance, but we do see a shift. The sigmoid colon, we see a much more sustained shift. Our EGTA assay, so we see the decrease and then recovery. Decrease and then recovery. And then what we're doing now is looking at different inflammatory markers and being able to look at how we could add different microbiome samples, similar to what we've done previously with the other two. So in the last couple of minutes I just want to tell you about our 2 other device types. I've not left very much time for this, but that's because the bulk of the work I'm showing here is published. We're working on this clearly now, but most of the work that I wanted to show you that was new has been on the in vitro side. So in terms of measuring gut motility, most of what we're doing is ex vivo using animal models, so rat or mouse, in organ baths where we can perfuse the tissue. And this is the type of device we've been using. So just a quick explainer on the enteric nervous system. So you've got these multiple layers in the gut. So you've got the central lumen. I've been focused on the mucosal layer here. But then you've got these 2 layers of muscle, and then you've got 2 components of the enteric nervous system. The myenteric plexus, which is really responsible for gut motility. But then you've got this submucosal plexus, which is more of that sensory neuronal function that I mentioned. The thing is they're intertwined, and the structure of the ENS is quite different from structure you would see in the CNS. It's a nerve net rather than what you see in the CNS. So it's more akin to what you would see in some of these animals like hydra or sea jellyfish, sea gooseberries, jellyfish, things like that. So it's highly complicated, which means that the task of understanding it becomes quite different. And also the design of the electrodes, you can't just use a multi-electrode array that you might use in the central nervous system. So in terms of measuring gut motility, we've used a fairly simple device, which is a strain gauge, meaning that when you stretch on this device, you decrease the resistance. So you stretch, you increase the resistance, and you contract, you decrease the resistance. And this has been well known. The trick here was that we needed to take our P.PSS devices, which I showed you earlier, and make them stretchable. And the stretch is quite large in the gut, so it can be up to 40%. So we had to show that that was possible. So a lot of material science went into the formulation. And what you're seeing here is a piece of gut with our device, which you can barely see, it's transparent, stitched or wrapped around, and then it's sutured, and so we're distending. This is our device. It's made by shadow masking, very simple protocol. And then this is the type of data, these are the type of data we get. So we're measuring the amplitude. So these are grouped neuronal signals called local field potentials. It's very characteristic of what you would expect to see. And then the strain, we can also measure that. So you see it's periodic. We can test this by checking stimulated or evoked responses, adding something like acetylcholine. And so if you're interested in this, you can check out the paper. We also did one piece of human stomach, which we had available to us. The final example is probably the most difficult. Again, this is published work. The electrode design is slightly different, but same, conducting polymer devices. The electrodes are these very small circles here. This is actually a suture hole, which was incredibly important for keeping the device in place. So you can see the device, or if this is going here, sitting on tissue, it's being distended, but you can see that there's a lot of heart movement, gut movement. So it's really important that the device stays in one place. There were a couple of things that needed to be figured out here. So the placement, so we placed it in the submucosa, in the colon. The development of that was pretty tricky, but also figuring out the anesthetics, because when you add anesthetics, you silence the neurons, so trying to get that right. So eventually isoflurane at 1.3%. And then there's more data in the paper that tells you more about what we did. One of the biggest challenges is in the deconvolution of the signal. How do you know which bits are the neuromuscular junctions, which bits are sensory neurons? So if you're interested, I encourage you to look. There we go. That's the paper there. It was a labor of love over many years. So Alex Boyes and Amparo led the work on that, but we had many collaborators who were essential to the work. So I'm going to finish there. I'd be happy to answer any questions. I appreciate I showed a lot of different things, but I hope I kept you with me. But maybe your questions will illustrate where I may have lost you at times. And then just final thank you to the Biochem Sup. Thank you.
Thank you, Róisín. As mentioned earlier, Professor Róisín Owens is the recipient of the 2025 AstraZeneca Award. Generously supported by AstraZeneca, this award is made every 3 years in recognition of outstanding work, which through biomedical advances leads to the development of a new reagent or methods. So at this point, we can now welcome questions for Róisín. So if you have a question, please type it in the question box as shown in the image on the screen. And I think we have a question. So first question from Samantha Gibbons. It says, for the chopstick electrodes, why do you not position them alongside the tissue? Is it because it is more convenient to get them through the junctions by positioning them above or below?
Well, it's because if you look at the chopstick electrodes, they're called chopstick electrodes because they resemble chopsticks. So the only surface really you'd be able to interface with the tissue is right on the tip. And they need fluid circulating around them. So the mismatch between the chopstick electrode in terms of stiffness, material, everything, it's like it's just a huge mismatch. So it just wouldn't work. There are different types. So the company who sells the chopstick electrodes, the lead company, they're looking at other ways of doing it. But fundamentally, it's a mismatch of the device to the soft nature of the tissue. I hope that answered your question.
Right, okay, thank you. So second question we have here from Ellis Baderewa. It says, interesting talk, just a quick question. Are the scaffolds measuring devices reusable, or are they typically used with a singular culture type, or how quickly can they be prepared for a new measurement?
So we can reuse them. The issue is getting the cells out of them requires some pretty strong denaturation protocols. But honestly, we can make the scaffolds at quite high throughput. So we're envisioning this as a disposable consumable, so a bit like a Transwell filter. The idea was that you'd be able to use them in culture for up to 5 or 6 weeks, so they would still provide a lot of value. So I think ideally they'd be disposable. There, by the way, they are autoclavable, so that's a nice feature.
Okay, right. Thank you, Róisín. Next question from Sophie Joblin says, you mentioned the richness and difficulty in analyzing electrophysiological data. What are the magnitudes of the measurements you are obtaining, and are there difficulties getting devices sensitive enough to measure small changes in, for example, ion flux?
Yeah, very good question. So I'd argue that if I go to the example of the quad culture model, so a vascular layer is really that very low resistance. So on a transepithelial resistance using chopstick electrodes, you can't see anything above noise. So it's in the order of 10 to 15 ohms centimeter squared. So that's a low signal. What happens when you approach your device to cells is you introduce a restriction on the ion flow, which means you're much, much more sensitive to the object you're trying to sense, which is the cell layer in this case. So in fact, we can measure the presence of cells which have no paracellular junctions at all. Now, it means you need a slightly different device type, and we've not really looked too much in it. But being able to measure the evolution of signal, which maybe I don't have that here, the evolution of signal. So if I started at day 0, I start to pick up that increase in signal from about day 3 or 4 before the cells are even confluent. So I'm actually super sensitive to what I'm trying to measure. Another point I was trying to make was that there are other things that you can measure. So I'll give you one quick example. We were looking at measuring lung barrier tissue. And by the way, we were doing that at the air-liquid interface, which we can do because the device sits on the mucus layer. So we've got an electrolyte. Otherwise, you wouldn't be able to do it. And we noticed an interesting signal here, which looked to us like a capacitance change. Now, capacitance often tells you something about the surface area. And we went back and did some imaging on the 2 samples we were measuring. And we discovered that the cells with the high capacitance that we'd measured had cilia, but the cells without didn't. And so we were able to pick up something, then we go and check, and it's a biological phenomenon. So there's other things you can pick out. The initial developers of this cell impedance sensing technique said they could measure micromotion. So I think lots more to do, but you have to validate everything with biochemical assays.
Thank you. Next question from Holly Hooper says, you mentioned the impact of microbes on resistance in the complex gut environment, what might be some limitations of conducting polymers?
I mean, maybe I'm biased, but I don't see a specific limitation of the conducting polymers. It's just a measurement tool. If you don't want it present as the scaffold all the way throughout the measurement, you could use our conformal devices just to do an acute measurement. The only thing I can think of to answer that question is that there is some evidence, not for bacteria, for viruses, that they may interact with sulfonate groups or materials. They may actually be antiviral in some cases, but I don't know a huge amount about it, you know. So if you were using the scaffold, could the viruses interact somehow? Maybe. We have one experiment set, it's a collaboration we've been having with a group here in pathology, looking at enteric viruses and our ability to detect those over time. And we've used the scaffold devices, and it seems to report on the virus infection quite nicely.
Right, thank you. So there's another question kind of related to polymers. It says, it was mentioned, this is from Grace Hepon, it was mentioned that the conducting polymer has a liquid formulation. Does this affect the storage of the polymer for further or future use?
Sort of. So the PDOT-PSS is actually a suspension in a liquid, and when you use it, when you buy it fresh, it's a really nice suspension, and then over time the PDOT-PSS, or the conducting part, may aggregate and sediment. And so when we store it on a stir plate. We tend to buy smaller bottles, so it can degrade over time. But once you've taken the liquid and either formed it into a thin film on those conformal devices or into a scaffold by freeze drying, then its structure is solid, no problem.
Right, thank you. And the last one I have here from Aruj Itisham asks, what are the main limitations of using fecal samples instead of real-time in vivo gut conditions?
So the fecal samples are a nicely available, fairly non-invasive sample. So a lot of these trials, they will collect a fecal sample. I think it's fairly standard of care. They can be stored for years at -80. It's though considered a human tissue act sample, so same as blood or tissue would be. So you have to go through that. You know, that's an important part of the storage. The worry that we had was that the bacteria wouldn't be viable, or whatever other microorganisms are in there, or that some of the metabolites would deteriorate over time. But other than that, I would say the fecal samples are very nice. Sorry, one other thing that's just occurred to me is that the fecal sample is a representative snapshot of what comes out. It's not going to necessarily be representative of the microbiome in the small intestine and even necessarily in the colon, because those samples have been exposed to air as well. So you hope it's giving you a snapshot, and maybe that changes from day to day or even hour to hour in the patient.
Right, thank you. So definitely the talks generated a lot of questions. So just a question from me is a kind of a generalist question. So you, at the beginning, you mentioned getting the balance between in vivo and in vitro systems, and there's almost like a trade-off between relevance and high throughput approach you use. So how do you advance the findings from these systems you are using to the kind of more relevant? So
yeah, really good question. So one of the ways I thought of was having a same parameter that you could measure in vivo, in vitro, ex vivo. So for example, when I showed the mouse gut sample where we could measure resistance using the same device as we'd used in vitro, I thought of that as a nice way to correlate our work in vitro with some work well, not in vivo, ex vivo. And you know, the future of measuring it in vivo would be, can we hook our device onto an endoscope and measure it in situ when somebody's getting an endoscopy? That's just one example, but I'll say something. So there's legislation brewing in the UK, in Europe, and in the US, where the FDA or the medicines regulators are looking at excluding animal testing from, for example, the first one will probably be monoclonal antibodies, and saying use in vitro plus in silico methods, because there's so much data out there already, and maybe the animal work doesn't really help us. So can we skip from in vitro directly to human? We're not necessarily super close to that, but there is a reinforced looking at the in vitro side of things. And so you could imagine there might be some large-scale screens, but then there could be some complex microphysiological models which are having more cell types, or thinking more about the gradients, or including microbiota, et cetera, et cetera, et cetera. And then combined with in silico modeling, can that really predict the safety or even the mechanism of certain compounds that are being looked at? So it's a very live question at the moment, I would say.
Lovely. Thank you very much. We appreciate that. So we would like to thank everyone for attending today's webinar, and a big thank you to Professor Róisín Owens for a fascinating talk. You can continue the conversation online. Follow the Biochemical Society. We're on Blue Sky, Instagram, or LinkedIn. We also welcome suggestions for future topics and speakers to feature in this Biochemistry Focus webinar series. If you have an idea for a webinar in 2026, we invite you to submit a proposal for an upcoming webinar. You can find more information about the webinars at biochemistry.org. All of our upcoming webinars are listed on our website as well. And if you have missed any of the over 100 webinars that we've already run as part of the series, or you would like to watch them again, please visit our website or YouTube channel. The recording from today's webinar will also be available to watch within the next couple of weeks. And finally, I want to highlight that it's ever more important that we stay connected and engage with your fellow molecular bioscientists. We have tailored membership offerings for all career stages, from students, early career researchers, through to established scientists. For me personally, the society has created opportunities for networking and collaborating with other researchers beyond my niche area across the broad area of protein-DNA interactions. So we really encourage you to join the society's community of researchers and specialists to engage with not only cutting-edge science, but advance your career development, grow international collaborations. So if you want to find out more, please visit the website and you get more information. And thanks to everyone, and goodbye.